This is part 2 of the video lecture on chapter 14 for microbiology. And we're starting with different cell types in the bloodstream. And we already talked about white blood cells, leukocytes, and now we're talking about erythrocytes and platelets. Erythrocytes and platelets per se do not play a role in immune function. However, they are important. Erythrocytes are just very simple cells. They are biconcave, so they look kind of like Mentos, I think, red Mentos, and they contain hemoglobin, and this carries oxygen and carbon dioxide to and from cells. Platelets are just very, very small blue-gray cells. They have no nucleus, and they release clotting factors, and they mediate clotting and inflammation. Okay. And here's an example of an erythrocyte, or examples of erythrocytes. You can see they're biconcave, and they are nucleated, but they're primarily used in and carrying oxygen and CO2. Platelets are these smaller cells here, okay, and they're involved in, I'm sorry, all of these are platelets, and these are burst platelets. Platelets are very sensitive to cuts and abrasions. You want them to be sensitive, so they actually burst open to release clotting factors, and they also release inflammatory factors. Okay, so if we look at the inflammatory response, so just break it down into how it looks in terms of signs. And then first we have rubor. Rubor means the color of the response, which is usually red. Calor, which sounds like color, which makes it a little confusing, but calor is heat. You actually feel heat at the inflammatory site. Okay, tumor, just like it sounds, it's like a tumor, only it's not a cancerous tumor. It's just something that happens because of swelling and loss of function. So the site of inflammation swells up, and then dolor, pain. Okay, so you've got pain in your response, and please remember those for the final exam: rubor, choler, tumor, and pain or dolor. Those are good terms to know. Okay, so if we look at inflammatory response, you'll see some different types of reactions that happen. Immediately, let's say that you get stuck with a nail, and the nail then injects the wound with bacteria. Okay, and initially, in order to prevent spread of the bacteria, you have to get vasoconstriction. Okay, so vasoconstriction lowers blood flow in this particular instance, and mast cells then start to release chemical mediators. Okay, then at this point, you get vascular reactions. It's the second frame. You start to get clot formation. Okay, clot formation then prevents blood flow. Clot formation actually can trap bacteria within the clot and prevent some of this bacteria from invading the rest of the body. And the bacteria are starting to move into the surrounding reticular endothelial space. Okay, and then neutrophils appear on the scene, and the neutrophils will then phagocytose these bacterial cells. Okay, and then vasodilation. Then with vasodilation, that allows then the different factors, different clotting factors, different inflammatory factors to seep into the wound site. Okay. And then 3, you get edema and pus formation. Okay, so pus starts to form in this yellow area. Okay, and this is due to the collection of fluid as well as immune cells. You get neutrophils. The neutrophils are starting to ingest the bacteria. Okay, and then fibrous exudate. You actually get a mat of fibers down at the bottom of the wound. Okay, and then finally a scab is formed in frame 3, and then finally in frame 4 you have scar formation. Lymphocytes then are coming along, and macrophages are being released through the bloodstream, okay, through the capillary walls, and they're able, like the neutrophils, they're able to phagocytose these bacterial cells. Okay, so vascular changes. Interestingly enough, you get constriction then followed by dilation, depending on where you are in terms of the stage of inflammation. Okay, this is controlled by the nervous system, and it's also controlled chemically by different mediators and cytokines. Okay, you have mediators that are vasoactive that can cause the smooth muscle cells on blood vessels to constrict as well as to dilate. And then you have chemokines that are chemotactic that will actually attract white blood cells. Okay, and then you have vasodilation, which will increase the flood of blood to allow immune components, immune cells, and antibodies to come into the wound site. Edema, we've talked about before. This is just when fluid starts to exude into the extracellular spaces. Okay, and the exudate is just the fluid that escapes from the circulatory system and the lymphatic system into the tissues. And this causes local swelling and also hardness of tissues just because of the presence of the exudate in the tissues. Okay, exudate can be clear, serous, or it can contain pus. When it contains pus, it basically has a mixture of white blood cells in it. Okay, so white blood cells and other debris are caused by phagocytosis, or debris, excuse me, caused by phagocytosis in addition to white blood cells. And here's an example of a wound where edema has developed. You can see rubor, which is this red color here, and you can see that the edema and the swelling, this type of edema is called lymphedema, and somebody's taken their finger and made an indentation in this inflamed area, and the indentation stays. Okay, the tissue is hard, and eventually this indentation will go away as tissues are replaced, or as fluid is replaced, but the tissue becomes hardened. Okay, and here you can see also rubor, which is the red color here, and you can see some exudate coming out. This is primarily serous, but there might be some pus in there as well. Okay, there's lots and lots of pus. This is a pyogenic wound. Pyogenic means that there's pus-forming bacteria in it, and pus again is white blood cells and debris caused by phagocytosis. Okay, so we'll look back at white blood cells. And when the white blood cells move out of the blood vessels into the tissue spaces, they transmigrate. This is called diaphoresis. Okay, and then chemotaxis occurs after these move out of the blood vessels into the tissue. The cells will migrate in response to specific chemokines, chemicals, and these chemicals are released at the site of infection. So we want edema and chemotaxis, at least in the short term, so we can provide fluid to flush away bacteria. We can also use chemotaxis to get white blood cells on the scene, and the white blood cells then can attack the foreign invaders. Okay. The influx of fluid dilutes the toxic substances. So if you have any toxins, if you have endotoxin from LPS, that will dilute it and cause the effect to be lessened. Okay. The fibrin clot, remember fibrin is the primary protein that's involved in clot formation. It traps the microbes, prevents further spread. And then neutrophils are immediately on the scene. Neutrophils are the most ubiquitous white blood cell in the body, and they phagocytose and destroy the bacteria, get rid of dead tissues and particulate matter. okay? And then if you have pus-forming bacteria like Streptococcus pyogenes, which we cultured in the lab, then we call this pyogenic, and you start to get pus formation. Okay, to look more detailed, take a more detailed look at phagocytosis. When you have phagocytic cells, then these phagocytic cells are just continually roving tissue compartments, and they're looking for microbes, particulates, even injured and dead human cells, and they do a housekeeping function. They ingest and eliminate these materials, and then they extract immunogenic information. They actually take cell surface proteins off of the bacteria or other pathogens and process that information so they can alert the immune system. Okay. The main types include neutrophils, monocytes, and macrophages. Okay. Neutrophils, like I said before, are the most abundant cell, white blood cell in the body. They are early responders. Once the inflammation occurs, they're ubiquitous in the wound site already, and so they start to react and phagocytose any type of foreign invader. Okay. Neutrophils are the primary component in pus. They're white blood cells, so the pus appears yellow-white. And you do get eosinophils. These are more involved in eukaryotic infections and not necessarily prokaryotic infections. They play only a minor role. Okay, here's a neutrophil. And this is a close-up of an eosinophil. Okay, monocytes are transformed into macrophages as they leave the blood vessels and go into the tissues. This is done by signaling molecules called inflammatory mediators. Macrophages are larger in size, and they have lysosomes. These are organelles that are developed in order to lyse bacteria, and also some other organelles that contain toxic components so you can kill the bacteria. And then you have what are called histiocytes. Histiocytes are macrophages that migrate and actually stay in a particular organ for their lifetime. So histiocytes are organ specific. Okay. And so if you look at the maturation of a macrophage, you start out with a stem cell. Okay, the stem cell makes a promonocyte. The promonocyte then migrates from the marrow into the bloodstream where it becomes a monocyte. The monocyte then undergoes diaphoresis. When it leaves the blood vessel, it goes into tissues, and then inflammatory mediators cause this to mature into a macrophage. Okay, and you can see phagocytosis. I'm not going to demonstrate that directly, but if you're interested in the animation, here's a website that you can visit. Okay, and if we look at the different stages of phagocytosis, this includes first chemotaxis by the phagocyte. Okay, the phagocytic cell is attracted by chemokines. Chemokines are attractive molecules that cause the phagocyte to migrate to the wound bed. Okay, the phagocytes then adhere to the bacteria via pathogen-associated molecular patterns. These are just molecules on the surface of the bacteria. We call them PAMPs. And the bacteria PAMPs then interact directly with cell surface receptors on the phagocyte. Okay. Then once the phagocyte starts to do its job, it engulfs the bacteria into the phagocytic vacuole. So you get a big vacuole that contains the bacteria itself. That forms what's called a phagosome. A phagosome is when a phagocyte has bacteria enveloped in it already. And then as the phagocyte then lyses the bacteria, you get fusion of the phagosome to a lysosome, so you get what's called a phagolysosome. Once it fuses, then mediators like lysozyme, break down the cell wall of the bacteria, release the contents, and kill the bacteria. Okay, and then once the cells are killed, then you don't want to leave around debris. Debris can cause a prolonged inflammation response, which is not good for the body. So the cells are completely destroyed and broken down into molecular components. Okay, and then any residual debris, the complement system can clean up afterwards. Okay, and here's a phagocyte. This is a large macrophage. You can see it's right on the surface of a blood vessel and starting to phagocytose bacteria. Okay. In terms of how the phagocytes work, they work by chemotaxis, and once they arrive on the scene, they can start to ingest bacteria. So the phagocytes are actually attracted by a gradient, a concentration gradient, that as you get closer to the parasite and the affected host tissue at the site of injury, once you get closer, then that gradient gets larger, so they're attracted by higher concentrations of these different stimulant products, and that will move the phagocytes to the site of injury. And then the PAMPs, again, they're on microbes, and they serve as red flags to the phagocytes and other cells in innate immunity. Okay, PAMPs are not involved in acquired immunity, so this is the second arm of the body's response. You've got the first line of defense, second line of defense. Second line of defense is non-specific immune cells and molecules, and so this falls in the second line of defense. Okay, PAMPs are innately recognized by the immune system. They are not a part of the acquired immunity. Okay, this includes things like peptidoglycan for both gram-negative and gram-positive cells, LPS, lipopolysaccharide for gram-negative cells, and other cell surface receptors. Okay, and then on the phagocytes themselves, there are what are called pattern recognition receptors. These recognize the PAMPs. They're already on the immune cell surfaces, and they bind the PAMPs directly. Okay, once the receptors bind PAMPs, then that sets in motion a defense response where the bacterial cells will be ingested, and the phagocytes then trap the microbes in the phagosome, and that's just a large vacuole that envelops the bacteria. Okay, and so when you have a macrophage and it starts to see a foreign molecule, okay, foreign molecule like peptidoglycan or a lipopolysaccharide, then you have these pattern recognition receptors. These are called toll-like receptors. They look like gates in a toll booth, okay, and they clamp down on the foreign molecule. And then once that happens, it initiates a cascade of protein signaling. So then you get cytokines, interleukins, and other inflammatory mediators that will stimulate cells to grow and mature, and cells to invade the wound site or the site of inflammation. And you also get inflammatory mediators that will cause the process of inflammation to be robust and flush away the microbes. Okay. Okay, then after the phagosome forms, then lysosomes are recruited to lyse the bacteria. And so the lysosomes fuse with the phagosomes, and that causes phagolysosome formation. Okay. So the lysosomes fuse with the phagosomes, like I said before, and then granules with antimicrobial chemicals are released, and these poison and dismantle the foreign invaders. Okay. This can happen within 30 minutes after contacting the bacteria, so killing starts very, very quickly after contact. And here's a phagolysosome, and you can see the lysosomes are much smaller. They're being recruited into the phagolysosome, so multiple lysosomes are being recruited, secreting their contents, and then killing the bacteria. And you can see the granules from the lysosomes being released. When that happens, then we have oxidizing agents like halogen ions, like fluoride and chloride, things like that, will oxidize the bacteria. Hydrogen peroxide, which is sequestered into the phagolysosome. It's toxic to cells. Superoxide anion and free radicals, all these are toxic to cells. Then this will cause lactic acid to be liberated. Lysozyme, which is the enzyme that breaks down peptidoglycan, and nitric oxide. And this not only kills bacteria, but it will also inhibit viral replication. So it is good against viruses as well. Okay. And then cationic proteins then injure the bacterial membranes. Okay, the bacterial membranes are phospholipids, so they're anionic. They have anionic heads, and the cationic proteins then will disrupt them. Okay, we have exocytosis. Once the bacteria or other invaders have been destroyed, then they're ejected. The debris is ejected from the cell. Okay. Now, when you have a fever is can be classified as a sign or a symptom. Okay, you can measure a fever, and that's a sign, and you can feel a fever, and that's a symptom. So this is just an abnormal elevation in body temperature. Okay, and this is when pyrogens are in the foreign invaders themselves. They reset the hypothalamic from the hypothalamus, thermostat to a higher temperature. Okay, and these pyrogens then elevate the temperature. These are exogenous. They come from the outside of the body. These can be from viruses. They can be from bacteria, protozoa, fungi. Okay. Endogenous pyrogens come from immune cells. Okay, and they're just a part of the immune response. So both the foreign invaders as well as the immune system can reset the hypothalamus to a higher temperature. Okay. Interleukin 1 and TNF, tumor necrosis factor, can reset the hypothalamus to a higher temperature. Okay, but you have to look at fever carefully, okay, because when you have bacteria, especially that are temperature sensitive, that can't replicate at higher temperatures, then fever actually inhibits their replication. So start to decrease the level of infection. Okay. It also, fever reduces the availability of iron, so bacteria can't scavenge iron from red blood cells. And fever also speeds up metabolism, and as metabolism goes faster, that stimulates the immune reactions, as well as naturally protective physiological processes. When you increase metabolism, you have a higher rate of flushing the organism from your system. Okay, so you have to be careful. You don't necessarily want to treat low-grade fevers. There's a variety of opinions on what temperature you need to treat in terms of treatment of fever, but you want to avoid high and prolonged fevers. High temperatures upwards to 105, 106 then start to destroy neurological tissues, and long fevers, long low-grade fevers can also cause destruction of tissues. Fevers in patients with cardiovascular disease, seizures, and other respiratory ailments are risky, so any type of fever in these patients needs to be treated immediately. Okay. Aspirin, acetaminophen, even ibuprofen, they lower the setting of the hypothalamus. My choice is ibuprofen. I think it's the safest alternative that you can use. Okay. You can also use cold compresses, cold baths can lower a fever if it becomes too high. Okay. And now let's talk about antimicrobial proteins, specifically interferon. Cells produce interferon. This is a small protein, and it's produced by certain white blood cells and tissue cells. and it defends against viruses and other microbes. It plays a role in immune regulation and immune intercommunication. And interferon alpha and interferon beta both will stimulate phagocytes. Okay, so they will allow phagocytes to enter the infected regions and start to phagocytose bacterial cells. Okay, and interferon gamma is produced by T cells, and that actually regulates macrophages, T and B cells as well. Okay, so when you have some type of cell that is infected, okay, then this cell, usually once a cell is infected with a virus, then it will be sacrificed. It will be killed by T cells. It will be killed by what's called T killer cells. The viral nucleic acids will stimulate the interferon gene. The interferon gene, once it's turned on, then will start to synthesize interferon protein. These are secreted from the cell, and then they go to a specific receptor on a nearby cell. This is just a bystander cell that isn't quite as infected with viruses, or maybe not infected, and this causes a specific signaling. And interferon, when it binds with its receptor, will signal antiviral proteins to be synthesized, and once they're synthesized, they'll break down the virus. Okay, so interferon is like an early warning system for the cells, and that allows maybe one cell will be sacrificed, or several cells will be sacrificed, but the bystander cells that aren't quite as infected then will have a chance to survive, and cells that aren't infected at all then will have defenses against viruses as well. Okay, so interferon also instructs the development of white blood cells. So as white blood cells mature, interferon plays a role. Interferon alpha activates natural killer cells. Beta helps in maturation of T and B cells and aids in inflammation. Gamma is also used in cancer therapy. It inhibits cancer cells, it stimulates B cells, activates macrophages, and enhances the effectiveness of phagocytosis. Okay. Then you have a whole another portion of the immune system called complement. And what this does is it actually complements, okay, for lack of a better term, immune reactions. And this is a network of over 26 blood proteins that will work in concert to destroy bacteria and certain viruses. This works in concert. All of these complement proteins work in a cascade. So if you have a small reaction in the complement cascade, then it leads to an amplified large response. Okay, well, like I said, cascade reaction. Then you get a physiological response where the first substance activates the next, and so on and so on. And as this cascade occurs, the reaction will be amplified. Okay, and there are three different complement pathways. There's what's called classical pathway, there's what's called the lectin pathway, and then finally there's what's called the alternative pathway. Okay, these pathways, the classical pathway is activated by the presence of antibody that's bound, and a specific antibody bound to the microorganism. The lectin pathway is when the host serum protein, this is just a blood protein, binds a mannan, which is a sugar that you find on the wall of fungus and some other microbes. Okay, and then you have an alternative pathway where complement protein binds a normal cell wall and the surface components of microbes. Okay, so there's three ways that this can be activated. Okay, and each pathway will ultimately lead to what's called a membrane attack complex, and the membrane attack complex actually pokes holes into the cell membrane of the invading bacteria or invading protozoa or whatever, fungi, whatever. And you can see that this is convoluted. There are a lot of different activators. Okay, classical pathway is activated by antibodies. Okay, the lectin pathway is activated by the presence of mannans, which you find specifically on fungus. And then the alternative pathway, which is slower and less efficient, will detect bacterial or fungal cell walls. It can also detect viruses and the surface of parasites like protozoa and helminths. Okay, and then this forms a particular complex. This then activates a cascade, and the cascade then leads to a membrane attack complex. Okay, so you get initiation. This is just a binding phenomena that initiates the complement cascade via one of the three pathways. And then the initial signal is amplified and the cascade occurs. The complement proteins then polymerize. They create a membrane attack complex, which is a polymer. And then once this polymer has been formed, then it can initiate membrane attack. And this complex then can digest holes in the cell membrane of the pathogens. Okay, now this is a really busy slide, but what you have here is you just have a pathogen. Pathogen is activated by C1. Okay, C1 then recruits C2 and C4. Okay, and these are active forms A and B of C4, active forms A and B of C2. Okay, and they also can be recruited by C3. Okay, C3 is broken down by an enzyme that splits these components. Okay, and then C3b combines with fragments of C2 and C4. This activates C5. C5 is then activated, and that recruits the rest of the complex. This is a membrane attack complex where you have a collar of C9 proteins, and then you have it's studded. It looks like a ring with a diamond on it. It's studded with C8, C7, C5b, and C6. And so this membrane attack complex goes back to the cell, okay, and it then binds to the cell membrane, and then it digests the cell membrane, and then it pumps it full of water. That causes lysis of the cell, and then the complement activation has done its job. Okay. You also have iron binding proteins, which scavenge iron. Bacteria love iron. They need iron to survive. So if you can scavenge iron away from the bacteria, then you will be able to limit the bacteria growth. There's also antimicrobial peptides. Now, iron is not generally available in the body, so it becomes a rate-limiting factor in the growth of pathogenic bacteria. So you have proteins like hemoglobin, transferrin, lactoferrin, ferritin, and they use iron for physiological reactions, but they keep it away from the bacteria. Okay. There are also antimicrobial peptides. The antimicrobial peptides are anywhere between 12 and 50 amino acids in length. They can insert themselves into the prokaryotic plasma membrane. This is the cell membrane, and disrupt the membrane in the same way, very similar to what you see in complement activation. And when they do disrupt the cell membrane, then the cell loses integrity and dies. Okay, and that concludes chapter 14. With this, I will conclude the lecture on chapter 14. And this will be the last chapter that we cover this semester. And then on Thursday of next week, we will review for the final exam.